Hello everyone, my name is Jay, and before we begin, I would like to ask everyone to please close their eyes. I want you to open your eyes and imagine seeing Earth in its entirety in your view. That's right, you are in space, floating, weightless, not a worry in the world, or not. It's a well-known fact that astronauts returning from their long voyages have many physical changes to their body. One of the less apparent changes occurs at the skeletal system, osteoporosis to be specific. But one does not need to go to space for skeletal system deterioration to occur. Limited weight bearing and bed rest can mimic the effects of anti-gravity conditions. Today, with the help of my peers, we are going to discuss how one can develop osteoporosis here on Earth as if they were in space.
Bone loss in spaceflight is a well-known example of secondary osteoporosis. Many studies on astronauts have revealed microgravity environments induce consequences on the bone remodeling process. A decrease in bone mineral density is evident in lower extremity bones like the femur or the lumbar vertebrae due to the decrease in weight bearing in space. This decrease in bone mineral density compared to the values obtained pre-flight is supported by an increase in bone resorption markers, such as collagen breakdown products, and also a decrease in bone formation markers, such as osteocalcin. Overall, gravity plays an important role in bone remodeling and Wolff's law as bone adapts to the loading, and in this case unloading, which explains the bone loss in spaceflight. It is evident that astronauts are at high risk for secondary osteoporosis, and further research is needed to find remedies that counteract the associated pathologies of spaceflight.
Now let's talk about bone loss in relation to bed rest. Bed rest studies are the only reasonable options we have to simulate microgravity, because going to space is not a destination you can just put on Google Maps and then just drive to. The subjects of the studies are performing all functional activities like eating and sleeping within the confines of a bed that is placed at a slight 6-degree angle. This causes blood flow that is normally going from your head to your toe. Now it's going to your toe to your head. The people that were involved in these types of studies were shown to have decreased strength in the knees and hip. In addition, shrinkage in muscle size was also shown within the muscles butt, thigh, calves, and knees. Another study showed bone density to decrease in cancellous bone and cortical bone. These are the two types of bone types in the body. Ultimately, bed rest studies emulate the effects of space flight microgravity strongly and the bone loss associated with it.
One way to obtain microgravity-based research is the use of clinostats, and clinostats are basically devices put around the body to eliminate gravity. And so a limited number of cell studies were done using clinostats to see the effects of bone loss. On one hand, we had some studies showing that cell morphology remained unchanged, but they still had a decrease in bone formation. On the other hand, these studies showed that gene expression for bone formation decreased, so indicating changes to gene level. So based on these studies, we can imply that genetics contribute to the greatest risk of osteoporosis development. But since some of the studies are still contradictory, it's important to aim future genetic research towards how genes regulate bone mass, specifically for osteoporosis. And by doing so, we can have preventive therapy for those at greatest risk.
Hind limb unloading was done on rodents to determine the effects of bone mineral density and distribution of cephalic fluids on the hind limbs. These results were then compared to those of astronauts after spaceflight. The structural and functional similarities and differences are used to find exactly what the effects of spaceflight travel are. In the study of rodents, bone mineral density showed a reduction from the hind limb unloading. The experimental setup took the load bearing off the back legs and lumbar vertebrae while keeping the load on the front legs and rest of the body. There were reductions in trabecular bone mass, volume, and osteoblasts, which are the bone cells that produce more bone. In the rodents, these reductions were found in the tibia and lumbar vertebrae, but not the rest of the skeleton. Cephalic fluid also shifted from the inferior of the body superiorly. These effects of reduced bone mass, volume, osteoblast count, and cephalic fluid shift are similar findings to astronauts after spaceflight. The major difference being that for humans in spaceflight is that their entire body experiences these effects.
The findings of the hind limb study described by Kyle closely mimics what is seen in individuals with osteoporosis. Osteoporosis causes a decrease in bone mass and calcium and altered bone architecture, leading to an increase in the risk of fractures. It is largely a phenomenon in the lower limbs where there's less weight bearing, and that's comparable to the study where weight bearing was taken out for rodents. The study revealed that when no weight is bared on the lower extremity, cells that are important for bone reabsorption and formation were reduced, leading to weaker bones. Once weight bearing went back to normal, the rates of bone formation and strength began to rise. Therefore, sustaining your bone mass and movement is extremely important because this will lead to increased bone strength, bone thickness, and bone mass. Regular exercise will better conserve your bone tissues, making them stronger and lowering your risk for fractures.
The article concluded by bringing attention to the roadblocks astronauts face for extended space missions, with bone density loss being one of the top issues. It also highlighted the possibility of using pharmaceutical companies to create drugs to target specific genes for this condition. As mentioned by my peers, bone loss in space mirrors many of the physiological changes seen in patients with secondary osteoporosis and those patients placed on bed rest. The other two areas of study showed how hind limb unloading led to bone density loss and how the use of clinostat machines facilitates anti-gravity effects on the cellular level for bone mass loss. Overall, more research needs to be done to best combat bone density loss, but the preventative benefits of staying active for patients with osteoporosis is clear. If you take one thing away from this, remember, for bones and muscles, use it or lose it.